Go. So, right, now that we've got some understanding, I trust, of how proteins are made and how amino acids are put together in the right order, now that we've got a handle on transcription and translation, there's a lot of things that we can see from our mountain peak now before we have to descend. So I talked about viruses. Let's talk about ways that bodies defend against them. The way humans and indeed most all vertebrates and even most invertebrates that I can think of, the way that we defend against infectious agents is with the immune system. By the way, any entity that makes you sick, a virus or a disease-causing bacterium, or even a disease-causing fungus, anything that makes you sick, is called a pathogen. I might use that term now and again. Pathos is Greek for suffering, and gen as in generate. So a pathogen is something that makes you suffer. And we use our immune system as a defense, except for the few unfortunate people who were born with no immune system, like David Vedder, who was born in 1972 and had to spend his life inside a sterile plastic bubble because he had no natural way to defend against viruses or bacteria. And knowing how the immune system works is critical for understanding how vaccines work. And I do not know what everybody's opinion on vaccines are. I'll actually be talking about that today and next Tuesday. Hopefully you can get a better understanding of exactly what they do and how they function, which might be useful in conversations over Thanksgiving dinner, maybe. So when we talk about your immune system, there's actually two immune systems that you've got, and one is the innate immune system. Think about the military for a minute. Let's say you're defending against a you're in a fort or something, and you're trying to defend against an enemy army. Some of your defenses, like cruise missiles or snipers with long-range rifles, have to be aimed and go after specific targets. But there's other defenses that that fort might have, like walls and trenches and barbed wire and, I don't know, machine guns on tripwires. Those defenses will act against anything. They don't have to be aimed. Once they are set up, they will continue to work. And that's kind of what the innate immune system is like. They don't have to be aimed at any particular intruder. And they include things like anatomical barriers. That's things like your skin. Inflammation is something we might be all too familiar with. You can get this even when you're not infected with a pathogen. It includes redness and swelling and tenderness and heat. That's actually part of the innate immune system. Fever is part of your innate immune system. It actually kills or discourages certain pathogens. Your body's white blood cells are part of the innate immune system, and so are some specialized proteins that your liver makes called the complement system. So if we look at these, your barrier defenses include things like your skin, which is pretty thick, it's pretty tough, and since it's always shedding and replacing its outermost layers, you can see this very clearly when you've got a sunburn, but at a lower level, it's doing this at all times. You're always flaking off the dead outermost layers of skin and replacing those with new layers. The toughness and the thickness and the constant outer shedding means that it's hard for bacteria to gain a foothold. I mean, sometimes they do, especially if you breach that defense and get them under your skin. Let's say if you get an infected cut or infected scratch or mosquito bite or something like that. But as long as that doesn't happen, your skin is a pretty tight defense against the entrance of bacteria. There are bacteria everywhere, including bacteria in your food. Most of them can get broken down by your stomach acid and the digestive enzymes that your stomach makes. Bacteria that might drift into your eyes can be stopped with an enzyme called lysozyme that breaks down bacteria, and it's present in your tears. Yeah, tears actually kill bacteria. The mucus in your respiratory system, this stuff, and this stuff I've still got a little too much of, traps bacteria that are breathed in on, in the air or on airborne dust. And it not only traps them, but if you cough it up or snort it out, the bacteria can be expelled before they can start growing. Which reminds me of the old joke about what's green and goes backwards at 100 miles an hour. Answer. Okay, sorry about that. Right. Inflammation tends to happen whenever you've got damaged cells. So you would see this if you had a bacterial infection in your body, but you'll also see this for non-bacterial injuries. It certainly happened to my knee back in February when I wrecked it for the first time. Whenever cells are damaged, they'll send out signal molecules, and that triggers increased blood flow, which makes the area turn red and feel hot. Increased blood vessel leakiness, and I'll talk about why that is in a minute, but your blood vessels get just a little bit more leaky, and fluid, hopefully not whole blood, but fluid oozes out of your blood vessels and causes the tissues to swell, and then increase sensitivity. So you get the 4 basic signs of inflammation: redness, heat, swelling, and pain. Or as the ancient Roman doctor Galen called it almost 2,000 years ago, rubor et calor et tumor et dolor. It rhymes in Latin, not so much in English. But those are certainly signs that everybody's familiar with. We've all suffered from inflammation at some point. And you'll get this whether it's a bacterial infection, a virus infection, or even just a physical injury. Fever seems to enhance other immune defenses, and it interferes with certain pathogens. Bacteria are often pretty finely tuned to survive in only a narrow range of temperatures, and you can slow them down if you get the temperature out of their comfort zone. I gather a number of physicians have argued that mild fever shouldn't be treated because it's your body's natural way of helping to fight off whatever's making you sick. Although obviously a super high fever can be damaging all on its own. But fever is part of the immune response. That inflammation process that we talked about triggers so-called white blood cells. These are kind of blobby, they don't have a fixed shape, and they travel through your bloodstream, but they can also crawl out of your blood vessels and travel freely in the body. They can get anywhere that they need to. And this is part of why your blood vessels get leaky in an area of inflammation. White blood cells can get out of the leaky blood vessels, travel to an infection site, and they can quite literally swallow living bacteria, dead bacteria, broken cell debris, or anything else. And because these cells don't have a fixed shape, they can just kind of attach to whatever they need to get rid of and engulf it, kind of flow around it. Like, I don't know if you ever saw the horror movie The Blob, but that's what it would look like. Just a huge shapeless blob flowing around you and completely trapping you inside. And then whatever is engulfed gets digested with more of these lysozyme enzymes that I mentioned earlier. So that process by which white blood cells engulf bacteria is called phagocytosis. Phago is Greek for to eat, and cyto is a cell. So phagocytosis means cell eating. Last but not least, you'll be thankful I didn't include a photo of this. Very large numbers of leukocytes surrounding an area of bacterial infection make up pus. Pus is basically this huge accumulation of white blood cells together with living and dead bacteria and busted bits of host cell and whatever else is there. Yeah, not that you really wanted to know that anyway, but that's what pus is. It's a sign your immune system is working well. An infection where you're not producing pus would actually be a lot more dangerous. And then finally, your liver makes over 30 specialized proteins that fit together to make up what we call the complement system. Some of the complement proteins attract leukocytes, white cells, and, quote, encourage them to do their job and engulf bacteria and all that. Others of them make up a set called the membrane attack complex. This is what they look like when they all fit together. All those orange blobs labeled C9, those are proteins that literally punch a hole in pathogens. These things blast a hole in bacterial cells and kill them. So yeah, these are, I don't know what you'd call these, kind of the landmines of the immune system. And they don't have to be specifically triggered. They'll work against anything. Okay, the adaptive immune system is what we've got to go through if we want to understand vaccines, because the adaptive immune system is tailored to respond to specific pathogens. So this would be like cruise missiles or, snipers or, smart bombs, guided missiles, things like that, things that are designed to respond to specific disease-causing organisms. And the adaptive immune system has a type of memory. The adaptive immune system can remember pathogens for years or sometimes decades. And that enables the immune system to mount a very fast immune response if it ever gets invaded again. The three parts of the adaptive immune system you need to know are two types of cell called T lymphocytes and B lymphocytes, and then proteins that they make called antibodies. All right, and yeah, before I took biology, I thought antibodies were like what an anthill was full of, right? If you kick an anthill, you can see all the little antibodies running around. But that was a joke. Right, anyway. Okay. You've got multiple types of white blood cell. And by the way, if you have an infection and your doctor takes a blood sample, the lab can gain some clues into what kind of infection it is by counting the numbers and types of different types of white blood cell in your bloodstream. If you've ever heard of getting a complete blood count or a CBC, that's what they're doing. And one type of blood cell, we've talked about the leukocytes. There's a type called macrophages, Greek for big eater, and a second type of wandering cell called a dendritic cell. And this purple blob here that looks like it's got leaves on it is a computer model of what a dendritic cell looks like. And macrophages and dendritic cells pick up pieces of whatever is triggering your immune system. Fragments of the bacteria, bits of the virus protein, pieces of the fungus or parasitic worm or whatever it might be. Any of those are called antigens. Please excuse me. Good to know my immune system is working well. And then these cells literally pick up the antigens and present them to a second type of cells, hand them to them, to a type of cells called T lymphocytes. And in this computer graphic, the T lymphocyte is shown in blue. T lymphocytes are found in a bodily system that you may not be aware that you've got. Hang on. Sorry, jumped the gun a little bit. Right, otherwise I'd die pretty quickly if I had no immune system. You've heard of your circulatory system. That's your heart and your blood vessels. You've got a second set of vessels traveling through the body. In this case, there isn't a special pump. The fluid in these vessels is just moved by overall muscle contractions. There's nothing like a heart. And it's a set of very thin-walled channels that link together lumps of tissue. The lumps are called lymph nodes, and the vessels that connect your lymph nodes are called lymphatics or lymph vessels. And the whole system is called the lymphatic system. If you've ever had, an infection, you might have noticed like a head or a throat infection, you might have noticed the doctor feeling the angle of your jaw, looking for swollen lymph nodes. Well, those lymph nodes are lumps of tissue that are part of the lymphatic system, and they're connected to each other by lymphatic vessels. And in that lymphatic system, we have T lymphocytes, E-I-E-I-O. Sorry, don't know what came over me there. And it turns out that you've actually got millions of different types of T lymphocytes. And what happens is something really weird. When your body is making T lymphocytes, there are particular portions of the genes that are shuffled randomly. And you generate these millions of different types of T lymphocytes, and each one is slightly genetically different from the others because portions of a set of genes called MLA get shuffled around when the cell is being produced. And you end up with millions of different T lymphocytes, each of which has a differently shaped receptor protein on its surface. This is what those receptor proteins look like. That's models of them. So you've got millions of different T lymphocytes. Each one has a randomly generated receptor protein, and each lymphocyte will make only that receptor protein for as long as it lives. Why does that matter? Well, this is what you'd see if we could shrink you down to about the size of a single protein. On the bottom, up on the top, you've got a cross section through a macrophage. This right here is the outer membrane of the cell. Let me shade that. Hang on. Yeah, this stuff that I'm shading yellow right there, for the moment, we don't really need to go into what it is and how it's made, but this is the outer membrane of that macrophage. And I'm shading in. Shade, 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 shade. And that macrophage is carrying an antigen. And this is it. This thing that I'm outlining in yellow, that's a bacterial protein or some kind of fragment of bacteria. And the macrophage goes around to T cells looking for one whose receptors fit the antigens. So on the bottom right here, what I'm shading in yellow right now is the outer membrane of the T cell. And sticking up out of this T cell right here, we have one of these randomly made receptors that just so happens to fit the antigen perfectly well. And so if the macrophage finds a T cell whose receptors happen to match those antigens, that receptor recognizes it and the T cell is triggered worse than a millennial whose Starbucks is out of almond milk, and it starts dividing. Sorry about the triggering joke. Right. So once the macrophage presents the antigen to the T cell starts dividing, and it produces two types of cells called cytotoxic Ts, also known as CD8 cells, or for short, killer Ts. Those cells go up against cancer and virus-infected cells. These will just walk right up to a cancer cell and break it open. They'll do the same for a virus-infected cell. Helper T cells are also produced, and these can stimulate the immune system response in various other ways. I'm not going to go into quite how. You can think of these as the cheerleaders. So when a macrophage presents an antigen to a T cell, the T cell will start dividing and it becomes either a killer T, which will go up against tumor cells and virus-infected cells and kill them, or it will produce a helper T, which can cheer on the immune system in various ways that I'm not going to get into. There is, however, another population of lymphocytes, not T cells, they're called B cells. And these do not need a macrophage to pick up an antigen. They just pick these up by themselves. And again, there are millions of different types of B cells, and they're created at random. When your body is making B cells, portions of their genes called, I think I said MLA, I was confused, it's HLA, portions of their genes get randomly shuffled. And so every individual B cell has differently shaped receptors. Every individual B cell has receptors that are made by this random process. And so you have millions of slightly different B cells in your body. And the B cells, again, B cells pick up antigens. And B cells whose receptors just happen, by luck, to fit the antigen start dividing repeatedly, and they turn into plasma cells. Plasma cells start cranking out protein like you wouldn't believe. Plasma cells just start going nuts and crank out a specific type of protein called an antibody. And that's what an antibody protein would look like. And antibody proteins have the same shape as those B cell receptors. So a B cell makes antibodies that match in shape the antigen that that B cell was given. Let me see if I can put that in a clearer way. A B cell that just happens to have the right shaped receptors. The right shape means that the receptors on the B cell match the shape of the antigen. And then the B cell makes antibodies that also match the shape of the antigen. So every one of your B cells can only make a single type of antibody. And you activate only the right B cell for the job when antigens come in contact with all of these B cells. Only the B cells whose receptors fit that antigen are stimulated to divide and start cranking out antibodies that also fit the antigen. In an in-person lecture, I'd be gauging my response by looking at your faces. I'm trying to make this as clear as I can. I'm getting a psychic signal that some of you might be a little bit confused, but bear with me for a second. What antibodies do is bind to antigens. And here again, it all comes down to protein shape. Antibodies bind to the antigens that their shape fits, right? Remember, we've talked about, like, screwdrivers fitting screw heads or sockets fitting nuts and bolts. Antibodies bind to antigens. There you've got a virus right there. In fact, I think that might be a COVID virus with a bunch of antibodies that are stuck to it. Those antibodies can directly interfere with what the virus or bacterium is doing. Antibodies trigger complement proteins, which come in and blow things up. And antibodies can physically clump bacteria together, stick them together in a big lump instead of letting them move freely. So they can physically interfere with what they're doing. And antibodies also stimulate phagocytes to devour whatever they're bound to. Antibodies basically say, yoo-hoo, Mr. White Blood Cell, over here, here's a nice tasty one, yoo-hoo. That process, by the way, is called opsonization. Opson, I think, is Greek for to have a meal. So opsonization is having a mealization because it's the process by which antibodies encourage leukocytes to engulf bacteria or anything else. You might recall, by the way, that one of the experimental therapies for COVID infection is, in the early days, you could take antibodies from a person that had already had COVID and inject them into a person that was sick. Using some really neat techniques that we just haven't got time for, these days it's actually possible to manufacture antibodies in a lab. And you might know that people that have full-blown COVID are able to receive antibody therapy. I think it's being sold under the name Regeneron. This is actually being tried for other diseases. My wife's mother came to live with us for several years because she has a type of cancer. It's called non-Hodgkin's lymphoma. And there was an experimental therapy available at UAMS that involved getting her injected with antibodies that were specially made in order to target the cancer cells. Because antibodies will also go up against cancer cells. It's not just the T cells on their own. And this was still experimental. It did work for her pretty well. I mean, she's still alive. That's the important thing. So there's a number of therapies that are out there and others that are coming into use now that involve manufacturing antibodies and injecting them into somebody that's sick to supplement the body's own antibodies. And that works, but gosh, it might be nice if you could make your own antibodies before you got infected. Well, it turns out you can. Some of the T cells and B cells become a very long-lived type of cell called memory cells, and memory cells can live for decades. Memory cells might last as long as you do. And memory cells just hang out. But if the body is ever hit with the same antigen, memory cells can divide quickly and produce new cytotoxic and helper T cells, and can also produce new B cells to crank out antibodies. So if the body's ever infected twice by the same virus or bacterium or what have you, the immune system remembers and is able to mount a response much more quickly. So this, for example, is why once you have had chicken pox, you don't get it again. You might get the chicken pox virus a second time, but memory cells make it possible for your immune system to fight off the chicken pox before you even know you've had it. So yeah, that's the immune system's memory of things that it has encountered before, is the memory cells. Memory, all alone in the immune system. La da 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 da. Anyway, incidentally, thanks to memory cells, breast milk contains antibodies to all the pathogens that the mother has ever been exposed to. If mom ever had chicken pox, then mom's milk ends up getting pumped full of antibodies to the chicken pox virus. If mom ever had measles, her breast milk is going to get full of antibodies to the measles virus. A newborn's immune system is still not fully up and running. And I really don't want to get into the debates about what you should feed your baby. People tend to get ugly about that sort of thing. But there is an advantage to women who can do it. Not everyone can, but women who can, their babies tend to be a bit less prone to illness because breastfeeding provides protection. The immune system of a newborn is still not fully up and running, but that breast milk, coating the baby's mouth and gut will protect that newborn against everything that mom has ever had. So yeah, there we go. And that's also the idea behind vaccination. If you give the immune system killed viruses or weakened viruses or broken up pieces of viruses or bacteria, etc., the immune system will quickly get rid of them. However, the memory cells can last for years afterwards. They might last for a lifetime. This was the idea behind smallpox inoculation. Oh, this is cool. This was actually discovered over 300 years ago. No, I take that back. It was discovered a lot farther back than that. But about 350 years ago, there was a Boston preacher with the odd name of Cotton Mather who had an African slave whose name was, we don't know his own name. The slave name was Onesimus. And Onesimus's people knew that if you took pus from a smallpox pustule and injected just a tiny bit into a little scratch into a healthy person's skin, that person would get a very mild case of smallpox, but would never suffer smallpox again. Getting one very mild infection would protect you against much more severe infections later in life. So yeah, that technique was actually introduced to America by a slave whose people in Africa already knew all about it. The Turks, if memory serves, also knew how this worked. We've improved on the technique a little bit because we can now take busted pieces of viruses or weakened viruses and do the same thing. You get injected with those. You may feel an immune system response. This is why you might get achy or, sore or, slightly ill when you get a vaccine. But the idea is that the memory cells that you produce will last a lifetime, and the immune system can respond very quickly if the real pathogen attacks. It's like we make your immune system play war games. There's a slightly added wrinkle with the SARS vaccines, because what the Pfizer vaccine is, you're not getting injected with virus protein, you're getting injected with messenger RNA that codes for one of the virus's proteins, not the complete virus. But one of the cool things about the DNA code is that it's universal. You could take DNA from any organism and put it into any other, and it could read it. So what you're getting is a shot of messenger RNA that causes your muscle cells to produce not a whole virus, but just a virus spike protein. And then your immune system mounts an immune response against that spike protein. And the idea is that the memory cells can last long enough that they can mount a quick response if you get infected with the real COVID virus. Ta-da! There we go. Yeah, the Pfizer and Moderna vaccines consist of messenger RNA. That codes for the virus protein. The virus protein stimulates the immune system, but it cannot actually make you sick. And no, the theory has been, not even theory, the accusation has been going around in certain circles that if you've been vaccinated, your body starts making COVID virus that you can shed and you can start infecting people if you had the vaccine. Not actually true. You're not getting enough messenger RNA to create a whole virus, just enough for one of the virus's proteins. The Johnson & Johnson vaccine is a crippled form of a different virus. It's called an adenovirus, and the adenovirus actually delivers DNA into your cells. That DNA gets transcribed and translated into coronavirus protein, and that coronavirus protein triggers your B cells to make antibodies. However, the adenovirus can't replicate in your cells. So, and again, this depends on the fact that that DNA code is universal. We could take some of your DNA and stick it into a bacterium, and the bacterium would start making your proteins, human proteins. Kind of, I don't know, like the coding standard for tape recorded music is universal. You could take I don't know if you even remember cassette tapes, you post millennials, but back when I was alive, we could take, cassette tapes and put them into a tape deck, and the tape recorder would play back whatever was on there, no matter what type of music it was, because all those cassettes had the music encoded in exactly the same way. Okay, that was me kind of flailing for a metaphor, but it's the same with life, that any organism can read any other organism's DNA. It's a universal genetic code, almost universal. There's some slight variations in it, but nothing we need to worry about here. Few more wrinkles to the immune system. There are times, especially in fall and spring, when your immune system is sensitized to attack something that's harmless, like pollen or peanuts or strawberries. And what you get is allergies. If you think about it, allergies, at least with me, I have I used to have a lot of allergies that were all, nasal. And allergies pretty much feel like colds. You get the inflammation response, right? You get swelling in your nasal tissues, so your passages get blocked up. You start cranking out mucus. You might spike a small fever. Allergies and colds feel pretty similar. Colds are caused by a virus. Allergies are caused by, something like pollen or mold, things like that can't actually make you sick, but the immune system is sensitive to them anyway. So yeah, allergies are basically the immune system overreacting to something that it doesn't technically need to. You can also have the immune system attacking, your immune system's got these cool ways of not attacking parts of your own body. Normally, you don't attack your own cells. The problem is the immune system will attack anything that is not you. And that doesn't matter whether that's a deadly bacterium or, in this case, a life-saving heart transplant. That's a heart being prepared to be being transplanted into somebody's chest. And your immune system cannot tell the difference between a heart that is going to save your life and a bacterium that's going to kill you. And that means that if you get a transplant, you have to dial back the immune system's response by taking immunosuppressive drugs. Taking immunosuppressive drugs is what keeps the body from rejecting and trying to destroy a transplanted organ, a heart or a liver or a kidney or what have you. The problem is it does give you a weakened immune system as a side effect, and you have to make some lifestyle changes if you get a transplant to make sure that you lower your risk of coming down with infections that you might not be able to handle. Before the development of immunosuppressive drugs, they started coming online in the 70s. And before they were developed, organ transplants were this science fiction-y stuff. Like the first kidney transplants were done in the 50s, and I think the 50s were when the first heart transplant was done. But not only is it rather difficult surgery, they didn't save lives for very long because people's bodies would start rejecting the transplanted organ, meaning that the immune system would start attacking it. And so what's made organ transplants possible, and what's made it possible for people to live for decades with a new heart or a new liver, is the development of drugs that block the body's own immune system, immunosuppressive drugs. You can also have the immune system overreacting. There are signaling molecules that stimulate the immune system that are called cytokines. And there are some viral infections that can cause cytokine storms, which is basically the equivalent of your body slamming every button on the control panel at once. The immune system's response is so strong that it causes severe collateral damage to the body itself. COVID-19, this is one of the ways that it kills you. You can die not so much from the virus itself as from damage sustained to your lungs by your body's overreaction. Kind of like trying to stop criminals in your city by bombing it from a B-52. Yes, it'll work, but you'll destroy most of the city in the process. That's kind of what's going on here, this overreaction in your body. And finally, this person right here has what's called a butterfly rash, those kind of two triangle-shaped red areas over her cheekbones, just under her eyes. And if you look carefully, you can see they kind of meet over the nose. That's called a butterfly rash. And it's a symptom of a disease called lupus. That's an autoimmune disease. Autoimmune diseases are what you get when the immune system gets its signals crossed and starts attacking a part of its own body. A good friend of mine has every so often attacks of rheumatoid arthritis. And that's the same thing. The immune system is going after cells in her joints. And you can live with this sort of thing, but you may have to take immunosuppressive drugs to manage an autoimmune disease. So autoimmune diseases, this would be like the police beating up innocent people instead of going after actual criminals. Unfortunately, we know that can never happen in the real world, but it's what your immune system is doing. Last thing, if memory cells last for many years, why the heck do they make you get a different flu shot every year? And what are all this worry about new variants of COVID-19? The Alpha variant and the Delta variant, and there's apparently a new one called the Delta Plus variant and so on. Where do these come from? Okay, when you duplicate your DNA, when that double helix unzips and then each strand becomes a template for building a new strand, what's doing the work is a set of enzymes called DNA polymerases. There's a second set of enzymes that repair damage to DNA. DNA is actually kind of a fragile molecule. It is pretty much certain that we're never going to clone dinosaurs because DNA is so fragile that even short pieces of it don't last the 65 million years that we need in order to get which is the age of the last of the dinos. So DNA gets damaged a lot and you have to repair it with another set of enzymes. And these polymerases and the repair enzymes are very, very, very good, but they're not perfect. Every so often they'll make a mistake and you'll end up with a T where there should be a G, or an A where there should be a C, or what have you. You'll end up with one of the nucleotides changing to something else. Sometimes this has no effect. Sometimes it may change the amino acid sequence and end up changing the shape of the protein. Sometimes it may even silence a gene and block it from being transcribed and translated in the first place. This can have effects that range from catastrophic to unnoticed. In eukaryotes, you also have, you might remember we talked about crossing over during meiosis. That's where chromosomes pair up and exchange pieces. Well, sometimes that doesn't happen perfectly. Whoever's sneezing, could you mute, please? All right. Sometimes that doesn't happen perfectly, and you can end up chromosomes that end up having a portion either deleted or duplicated. So you can have a portion of a gene that gets cut out, or a portion of a gene that gets a portion of a gene that gets repeated. You can even have entire genes or even whole sets of genes that either get cut out or get duplicated. All of these changes can be passed on to the next generation, and we call them mutations. And this is why you have to get a flu shot every year. Mutations are constantly creating new strains of flu. The flu that you got your shot for last year is going to be different from the variety of flu that is spreading this year. And, the World Health Organization tries to track what varieties of flu are likeliest to be a problem. So every year you get a flu shot that will help you make antibodies against the strains of flu that look like they're going to be common. There are times when predictions prove wrong, and you might get the flu shot and still get the flu if you're exposed to a virus that's different from the varieties that we thought you were going to get exposed to. That happens. But yeah, mutations are constantly generating new varieties of flu. It's also why we haven't developed an HIV vaccine yet. And HIV mutates very fast. In fact, it mutates even within one patient's body. If you get infected with HIV, within probably days, but certainly within months, you're going to be producing varieties of HIV that nobody's ever seen before, because HIV replicates fast and it mutates very frequently. And it mutates faster than the immune system can adapt. That's why full-blown AIDS is fatal. Is HIV produces new varieties within the body of an AIDS patient faster than the body's immune system can keep up. And it's why it has not been a simple process to develop an HIV vaccine. So just by way of example, what you're looking at in the center is a diagram of the alpha variant of COVID-19. In the center right here, I'm kind of shading it in. There is the, I think this is RNA. I could be wrong. Inside the capsid, and then the whole thing, the thing with the little spike proteins on it, that's the capsid. Surrounding that, you have a diagram of the alpha variant's DNA, and compared to the original version that was discovered, it's got several mutations in capsid proteins, as well as a couple of deletions, and then a bunch of mutations in the spike proteins. That's the part of DNA shaded in red. And those happen to make it more infectious. One of the mutations in that spike protein changes amino acid number 501, stops being an asparagine and starts being a tyrosine. That causes a small change in shape in the tip of the spike protein. And that change just happens to improve the virus's ability to bind to cell surface proteins. So that alpha variant was more infectious because it was better able to bind to cell surface proteins compared to the early COVID variants that we discovered back in December 2019 was when it first popped up on the radar, back in the before time. So yeah, sometimes a change in amino acid sequence does nothing. Sometimes it knocks the protein out completely. Sometimes it actually enables the protein to work more efficiently. There are probably lots and lots of variants of COVID that never got anywhere because the mutation made the spike protein less able to function, right? Mutations don't have to be good for you. They can be bad. And there've probably been a lot of mutant COVID viruses where the mutation made the spike protein fail to work, and we'd never even know they were there. But every so often you can get a mutation that does benefit, in this case, the virus. It certainly doesn't benefit people, but it makes the virus better able to bind to cell surface proteins and infect host cells. The Delta variant, okay, what you're looking at here is these lines right here are just represent lengths of virus DNA. The Alpha variant right here at a position called 69 to 70 has a deletion. And here at position 144, it has another deletion. And at these little dots right here, these are points in the DNA where one mutation has happened that changes an amino acid. The Beta variant has a different set. The Gamma variant has a different set, and the Delta variant has yet another set. Has a number of places where amino acids are altered compared to the first COVID that we found. And so there's one right there. There's a change there, there. There's a change and a deletion there and there and there and there. I'm just highlighting each one of them. Every one of these codes right here means a change in the identity of an amino acid at a particular position in the chain that folds into the spike protein. So yeah, this is kind of cool. We can compare spike proteins from different variants by literally lining them up right next to each other and seeing what specific amino acids have been altered. Incidentally, anything that damages DNA is called a mutagen, which just means something that causes mutations. So there are chemicals that can cause mutations. Radiation, getting, X-rays or gamma rays or something like that can cause a mutation. Ultraviolet light is very good at damaging DNA. All of these damaged DNA, sometimes that damage doesn't get repaired perfectly. You've got enzymes in your cells that fix DNA damage, but they don't always work. What you're looking at here in the picture is a portion of a plastic plate filled with a jello-like substance called agar. The white spots on top of the jelly are little colonies of bacteria. And what you can do is, if you've got a chemical that you think might be a mutagen, you can expose it to a bunch of bacteria and see if any of the bacteria develop mutations. And there's a fairly simple and cheap way to do this called the Ames test. The Ames test is a check to see whether a substance causes mutations in bacteria. And this is the first line test for mutagenic activity in newly discovered chemicals, even consumer products. If you have some new ingredient that you want to put into the next line of lip gloss or something, and you need to know whether it's going to cause cancer or not, there is still some animal testing that's involved. But the first test you do would involve exposing bacteria to this chemical and seeing how common mutations are in the bacteria. Because again, that DNA code is universal, and anything that makes bacterial DNA mutate has a pretty good chance of making your DNA mutate as well. And so it's because of mutations that we have new varieties of virus popping up. It's because of mutations that we have new varieties of bacteria popping up. And some of those mutations improve the virus's or the bacterium's ability to grow and reproduce and outwit the immune system. And those become more common. And some of those mutations decrease the bacteria's or virus's ability to dodge your immune system, and those become rare. And what we've got is, lo and behold, natural selection. This is why this stuff was important way back in about week 4 and 5. Now we've got a handle on something Darwin didn't fully understand, which is, where does variation come from? Well, a major source of where variation comes from is mutations. Probably most mutations are not harmful at all. Most of them probably have no effect, but every so often a mutation will have an effect for better or for worse. In the case of flu viruses or COVID viruses, some of those mutations don't do anything. Some of those mutations might decrease the virus's ability to replicate. And if that happens, then that type of virus will become rare and eventually go extinct. Or some of those mutations will improve the virus's ability to replicate, and viruses that have those mutations are going to become more and more and more common. And we end up with new strains of virus that are more efficient at affecting people, and possibly, although I haven't seen signs that it's really happened yet, possibly more efficient at getting around the defenses that vaccines can provide. This, by the way, is why it's still important to wear the freaking masks and get the bleeping vaccine. Every person who gets COVID may have, I don't know, billions of viruses inside that person's body. Every time one of those virus particles gets made, there is a small chance that that virus particle is going to have a mutation in its genetic material. And there's a small chance that that mutation is actually going to be more infective. It's a very small chance, but it isn't zero. The more people get infected with COVID, the greater the odds that some mutant version of COVID is going to pop up that is more efficient at infecting people, better at killing people, or better at dodging the defenses that we've got so far. That's why it's important to cut down on the total number of people that have COVID. The fewer people that have it, the lower the odds that we're going to get some mutant variety in the future that defeats us and knocks us back to where we were in March of 2020. Okay, right. That's a pretty good place to stop. So stopping is what I am going to do, and I will now stop the recording.